Bonjour, bienvenue à notre
Good day, and welcome to our fourth Zebinaire for the Quebec Coalition of Rare and Orphan Diseases on this month of February. And the theme this year is the Odyssey of People Without a Diagnosis. So the diagnostic wandering. My name is Gail Ouellet, President and Scientific Manager of the RQMO. And I would like to briefly introduce the RQMO. For those who don't know this group, our main mission is to inform and support people with a rare disease and also an undiagnosed disease. So in case of rare diseases, people can get information, resources, help, and referrals to the medical environment. We also do general education and information on rare diseases. Our center has helped some people find places in our healthcare network to get a diagnosis to support them during their diagnostic wandering journey. To give you a few statistics, based on the estimate of our Ministry of Health of Quebec is using, it is 7 to 8% of people in the population that would be affected by rare disease, which would be approximately 700,000 people in Quebec with close to 500,000 children. At this point in the journey of someone with a rare disease that is undiagnosed, 40 to 50% of these people at one point have no diagnosis. Some will get one and others will never get one during their whole life. A survey we made at the beginning of the foundation of our organization with more than 300 people with rare diseases and personal support workers for those people. Our respondents told us that 37% of them took 3 years or more to get their diagnosis, 18% took more than 10 years, and 30% had to see 5 physicians or more before getting their diagnosis. Those surveys look pretty similar to those around the world, even one recently done in Ireland. We're talking about an average that could be 7 to 8 years to get a diagnosis when you have a rare disease. Of course, doctors cannot learn all rare diseases. There is about 7,000 to 8,000 noted throughout the world. It's so in a way it's normal that it takes some time before being diagnosed. However, there are consequences to this diagnostic wandering, and there could be inappropriate treatments, and often treatments are done by trial and error because doctors don't know exactly the cause, the etiology of the diseases. There are false diagnoses that happen also during the years, and genetic diseases have consequences on absent or imprecise genetic counseling because of the absence of a cause for the disease. So families don't necessarily know the risks for their family members, or if they want to have another child. Psychosocial consequences are high. There's uncertainty, false hopes when you get several diagnoses that are invoked at different times, difficulty to access adequate services, especially for financial assistance, status as a disabled person, psychological distress. And when the wandering lasts for a long time, there's a loss of confidence in the medical system. We have proposed some solutions over the years after listening to what patients and natural helpers wish. It is clear that there is a need for our physicians and healthcare professionals in the network, not about the 7,000 rare diseases, but about how to approach rare diseases. What is the right diagnostic approach, understanding the obstacles and the barriers experienced by the families so that you understand what is diagnostic wandering from their point of view. So we ask for that type of training and resources that we need. Family physicians or urgentologists or other specialist physicians, they should know where to refer their patients so as to increase the likelihood that they get a diagnosis. We also look at an experiment that was done in France in the field of rare diseases. They speak about the culture of doubt. Physicians are encouraged to dare think about rare when they've done all their exams and they haven't found anything. During the month of February, we have testimonials on our Facebook page. Let me present two of them, which are related to the topic today. We have here Marie-Josée. It took 30 years before she got a diagnosis. She had a lot of signs. This was a multi-systemic disease, and it's thanks to the new sequencing technologies that a few years ago, a gene was identified in her case, and to our knowledge, and there are two remote cousins of hers that are affected and another person in another country. So those are really ultra rare diseases, almost unique diseases, single cases almost. And Thomas recently just got his diagnosis after 14 years. And there too, it's like a new disease associated with a specific gene, and the signs and symptoms of his disease were different from what people knew or believed. And it's thanks to the identification of a new gene that we could get to that diagnosis. So today, this will be the topic of our Zebinaire. Before introducing our speaker, I also wanted to thank our sponsors for all those activities that we're having during the month of February and the projects we have for our QMO. So I'm now going to introduce our speaker for today, who is Mr. Serge Rivest, a researcher and professor at the Department of Molecular Medicine at the Faculty of Medicine of Laval University and director of the Research Center of the CHU de Quebec at Laval University. He holds the Canada Research Chair in Neuroimmunology because actually his research work deals a lot with the regulation of the genes in the immunity system, in the central nervous system, and in particular, very exciting, he works on cellular therapy to treat neurodegenerative and demyelinizing diseases. And today we're going to listen to him because he's also the director of the Research and Innovation Center on Rare Diseases under the acronym PRISME, a Research and Innovation Program on Rare Diseases. It's a project about how to use new sequencing technologies in order to identify genes that are related to some rare diseases in order to provide diagnoses to people who are erring and wandering in search of a diagnosis. So thank you for being here, Dr. Rivest. I know you're very busy and we appreciate the fact that you're here today to present your project. So over to you, Dr. Rivest. Thank you very much. Can you see my presentation on the screen? Yes, we do. Okay, we're good. So today I'm going to present our project, which is about this group, which we call PRISME, which is the Research and Innovation Center on Rare Diseases. The objective of this project is to improve knowledge about the molecular functions related to some rare diseases, but our goal is mainly to identify the genetic causes associated with some rare diseases, such as neuromuscular syndromes, neurodegenerative syndromes, polymalformation, and metabolic syndromes in children, or young adults from Quebec, where the cause or the genetic factors involved has not yet been identified. We also want to get a better understanding of the molecular mechanisms involved in one of those 4 types of diseases that we are covering. So today, I really want to present to you our research projects on genetic variations. So I'll give you a brief summary of what is a genetic variation. There are several types of those. We can have, for example, chromosomic aberrations. Usually before being admitted to a project, patients undergo preliminary genetic tests to detect obvious aberrations in the chromosomes. So we at the SCHUL, what we do, we have children who have a disease, and we do tests, genetic screening tests, to see whether or not we can detect one thing or the other. A karyotype analysis examines all the chromosomes in a patient's cell from a microscopic view. This test assesses the number and structure of a person's chromosomes in order to detect chromosomal abnormalities such as trisomy 21 or Down syndrome. A normal karyotype contains 46 chromosomes arranged in 23 pairs, 22 of which are identical chromosomes, 2 by 2. And there's 1 pair of sex chromosomes, which can be XX or XY. The formula is written, for example, 46XX for a healthy woman and 46XY for a healthy man. You can see here the difference with a man and a woman. So that's where we do the first detection. There are some chromosomal aberrations also. For example, the first test we do with our children, we do a CGH, comparative genomic hybridization chip. Actually, we do this for most patients. This analysis compares the patient's genome with a reference genome, and it identifies differences between the 2 genomes and thus locates regions of genomic imbalance in the patient. We can see by hybridization here the DNA using a very specific probe that will give us a color. The color depends on the hybridization that occurs. Either it can be green or red. So by hybridizing with the probes that are green and red, we can detect chromosomal aberrations, or rather DNA aberrations. Types of mutations. There are mutations which we call point mutations, which can be false meaning, silent, or nonsense. And there are aberrations of insertion or deletion. There are point mutations, which we call alternative splicing, changing the splicing. There are regulatory point mutations and also unstable repetitive mutations or transitions. There are complex rearrangements. Now, a silent point mutation, which we call also, it's simply a mutation in which one nucleotide of a codon is changed, resulting in no change in the associated amino acid. So that's a silent mutation. There is not necessarily any major change in the DNA code, which encodes for proteins, as we know. And the proteins are what are not affected in these cases. So this is a mutation that will not have any major effect on the proteins produced by the cells. There is also what we call mutation point false. It's a mutation where the type of a codon is changed. It's a single base pair that is changed. It's very specific. This is a substitution. Here you have a substitution in the codon. You see here a change in one nucleotide that is changed. There is also genetic variations that could be nonsense point mutations. This is a mutation in which one nucleotide of a codon is changed, inducing the change of the associated amino acid to a stop codon. The resulting is a truncated protein, which is generally non-functional. That's where we detected disease with this non-functional protein. there are genetic variations with consequences in nonsense point mutation. For example, we'll see changes in the messenger RNA, which will be associated with a protein that will not be synthesized. And there will be a truncated protein that will be synthesized instead. Genetic variations can be the consequences of nonsense point mutations. These nonsense mutations are such that the protein will not be synthesized. There are genetic insertions and deletions with or without a frame shift, a lag in the reading code. So this means that a certain number of nucleotides in a sequence will be moved or translated to another point. Here you see the normal sequence of DNA, or an insertion mutation, non-frame shift. So that will produce a protein that will be truncated. There are genetic variations, which we call point splicing alternative mutations. That's the creation or the abolition of a new splicing. And this is what is demonstrated in some persons who will develop some of those diseases. There are regulatory point mutations also. Here, if I may say, this is a regulatory point which can have an effect on the promoter, and the promoter will regulate this mutation. There are 4 examples of regions that can affect the regulation of a gene. As we can see them here, it's due to the position of the gene, or what we call the promoter, the exon, and the intron. We can see in various sections of the DNA at what level these effects occur. There's an effect due to position, and there's a cis effect, which is an effect on the enhancer, and there are transcription effects, which we call the trans effect, and the promoter also, which will have a direct effect on the transcription or the blanking of an exon. Then there's unstable repeat expansions, which could be caused by a mechanism called a slippage during DNA replication and repair. That is totally different from what we presented before. It's due to the replication of the DNA. There are also several diseases linked to the repeat expansion, repetition of 3 nucleotides. Most of those are related to non-degenerative diseases. Here, there's a type of disease such as, for example, Huntington or myelic dystrophy or Friedreich ataxia or the Fragile X syndrome, and other diseases such as the Jacobson syndrome. So mutations can be of different types. There could be wild type that we can't really identify. It can be heterozygous, homozygous, and they could be either trans or cis. The modes of transmission of the mutations now. There is autosomal dominant. Then with each pregnancy, the risk that a child will be ill is 50% with this autosomal dominant transmission. There's another mode of transmission, which we call autosomal recessive. With each pregnancy, the risk that a child will be ill is 25% in these autosomal recessive mode. There's also what we call the dominant mode of transmission. Then with each pregnancy, the risk that a boy will be ill is 50%, and the risk that a girl will be ill also is 50%. With each with another type, the risk that a boy will be ill is 0%, but the risk that a girl will be sick is 100%. There is the X-linked recessive mode of transmission. There, with each pregnancy, the risk that a boy will be ill is 50%, and the risk that a girl will be a carrier is 50%. With each pregnancy at risk, the risk that a boy will be sick is only 0%, but the risk that a girl will be a carrier is 100%. There's the so-called de novo mode of transmission also. De novo is linked with the family history. For example, it could have an effect up to 30% and 50% could be hereditary. It can happen that a recent mutation, de novo or a neo mutation, as we call them, occurs in a sex cell of one of the two parents or both parents. If this mutation causes a dominant pathological allele, although neither parent is affected, one or more of their children may be affected and pass the mutation on to their offspring. Here we could summarize what we call the modes of transmission. One of the modes of transmission is the mitochondrial mode. A mitochondria is a particle that we find in a cell. Mitochondrial DNA is a small circular chromosome found inside the mitochondria. The genes located in the mitochondria are few in number, but mutations in these genes are very common. During fertilization, only the nucleus of the spermatozoon enters the egg. The contents of the cytoplasm of the egg cell are therefore the cytoplasmic contents of the egg. This is why all the mitochondria of an individual come from their mother. The severity of the disease depends on the proportion of an individual's mitochondria in which the gene involved in the disease is mutated. And that mutations are what I'm going to explain later on. Indeed, in each cell, there are between 500 and 2,000 mitochondria that divide autonomously within the cell, and these mitochondria are randomly distributed in the daughter cells during cell division. So, what is the methodology that we used? It's as follows. First, we recruit patients. We do the blood samples. We treat the samples which are deposited in a biobank. We sequence the exome or the whole genome. We do a variant analysis. We do further tests, and then we disclose the results to the families. The recruitment of patients occurs as follows. We take living patients and biologically related members, whether they're affected or not. We give priority to pediatric patients, but young adults may also be recruited. That everything is done here at the CHUL. They should have obtained the diagnosis of a neuromuscular pathology, neurodegenerative disease, polymalformative syndromes, or inborn errors of metabolism. They must not have had the cause of the disease identified by a clinical genetic analysis following reasonable investigation by the physician. So first, we do everything that can be done in the trials we do with gene panels, and when nothing is found, then the patient is recruited in our study. The patients also must have been identified as a potential participant by the supervising physician. In the vast majority of cases, we absolutely need to find the trio, the father, mother, and child. And if there are sisters or brothers, we also recruit them into the study. We need at least father, mother, and child. The methodology is as follows. We do the collecting of biological samples. We take the blood, we extract DNA, we immortalize the cells, and so as to have cells to do subsequent studies. If possible, we also sample urine. We extract proteins from the urine to proceed with the evaluation. We also take saliva. We extract saliva. We extract the DNA from the saliva for sequencing the exome. Western tests are done on the urine. And if there's a neuromuscular disease, we take a biopsy to see the DNA, the RNA of the protein, and we do analysis of their various tissues. So extraction is a classical DNA extraction that we do through a DNA extraction, as we usually do. And for the rest, we do a sequencing analysis. If we immortalize the B cells, why do we immortalize those B cells? Because we want to have cells to work on for the rest of the studies to do subsequent analyses if we found a variant. We do cytochemistries, for example, to deploy the cells and multiply them. And then we freeze these cells in liquid nitrogen for years to come. The sequencing of the exome is a sequencing where we amplify DNA and we proceed to sequencing by new devices called new generation biosequencers. Then, and that's where it becomes more complicated. How can we find a variant? Once we've done the sequencing of the exome and the genome, the level of information is extremely complex. So in our lab, we have developed together with biocomputer specialists and data engineers and data architects, what we call a pipeline of data. This is what we call the GATCK. It's a genome analysis toolkit, as we call it, the GATC. Then we proceed with variant analysis, and then we put this into our data machine to see what are the potential variants that could be found for some mutations. Then we'll find at that stage hundreds and hundreds and hundreds, if not thousands. How can we narrow it down to find the variant that causes the disease? That's where we analyze with hundreds and hundreds of data to find what is the factor that gives rise to a variant. It took years and years before demonstrating that. We had to collaborate with several researchers in the whole world, researchers from Vancouver and Ottawa, for example, to really find the variants and the base pairs. So the tools to help prioritize and interpret the variants, that's the most important thing. Otherwise, we'll never find. So the variants that we can find some that have already been identified. We go to the database to find the human genome tissues. In the database, we can find variants that have been previously identified, because the goal of our project is to have a big directory to offer to the whole world variants that have already been demonstrated. Then there's the frequency of variants in the control population. We can see aggregations in some consortiums. We can see exomes of variants or projects, midterm projects that already exist to help us interpret those variants. There's information about the gene, which we call sequences of positions that can be present, or the NCBI, the National Center for Biotechnologies, or you can find those in the genome sequencers. And we do all this research to help us find these variants. There's information about the gene also, which we call the function and expression of interaction with the disease. You have the midline, PureMed, the Gene Corp, Gene Mania, Gene Textbook, which is a portal that helps us find this balance and find this needle in a stack of hay. There's information also about protein. Protein information provides in silico predictions of the effect of changes in a different amino acid that will change position. These programs take into account the physicochemical difference between two amino acids, and will define whether the change is in a functional protein domain and whether it is evolutionarily conserved. Systems exist, which we call the human splicing finders. There's also a predict protein software and a whole lot of other systems through which we can do in silico analysis of proteins with the tissues that we have sampled. Then there's all the interactions with the existing systems, such as, for example, we collaborate with Ottawa on Care for Rare. All the other systems on which we can collaborate on to get a synergy between all the platforms that are currently existing with all the people working in rare diseases globally to possibly find matches. And that's what we show here. Here we can get matches to find those variants. And matches could be done with Vancouver or Ottawa, or it could be with other countries. We could go there to find those matches. And when we find such matches, then we'll find some evidence, the pathogenicity, the annotations, frequency, location, and quality. This is family 01069. This is a gene which we found, MFN2, and this is a family where we found a variant called missense. So this gene, we can do the nomenclature. What is it used for? What does it do? Where is it expressed? And are there other cases that were found about this in the whole world? And we also did what we call clinical confirmation of the variant of interest, because we can't go see the families and tell them that we found a variant, to tell them, okay, this is the variant that causes your child's disease. So we absolutely have to be able to confirm the mutation through a subsequent methodology. That's what we call the methodology by Sanger, which is compulsory in our case. As long as we made the confirmation, we are convinced that this is the gene that causes the disease. Only then can we contact the family. There are functional tests also. PCR cloning to do a functional test to really confirm that the gene does cause the mutation. We will also confirm this through a method we call the RT-PCR to make sure that this is the right variant. Then we will confirm that the variant will cause a dysfunctioning of the protein. And for this, to check this dysfunctioning of the protein, we have to use another technology called the Western blot. Through Western blot, we can demonstrate that there's actually a dysfunctioning of the protein caused by this variant. There are different other tests also. We want to do enzyme tests, transfection tests, and RNA-seq tests to show that other variants can cause other mutations. After that, once we're convinced that this variant does cause the disease, we then disclose the results to families, and that's often what's most difficult. Our research nurse, Mehdi, will go meet the families, and then we'll say, here's what we found in your child, and is there a drug or not? We put the information together in relation to participants at all steps, from recruitment to findings. We do the pedigree, the blood work, genetic tests, the phenotype, the CGH, biopsy, the variants found, sequencing, blood, karyotypes, resolved cases, and publication, as the case may be, immortalization of cells, DNA sequencing, and patient recruitment. So everything is put together in a very large database called REDCap. And that this REDCap database is shared with other research groups working on rare diseases. Project status now, where are we? So far, we have recruited 135 families. We have sequenced close to 500 exomes. We have solved 31 cases, so we saved the lives of several children. We did 5 publications so far. I'll present to you now 2 cases. Case number 1, it was a 6-year-old boy with muscle weakness, a motor delay during the first year of development. At the age of 6, he could not run, jump, and had difficulty climbing stairs. Clinical examination showed generalized muscle atrophy. Extensive genetic and clinical examinations were unsuccessful. So the participant and his family were then enrolled in the PRISME project. The person, the father included the father, mother, and sister who were also recruited into the project. The consanguinity of the parents suggests what we call an autosomal transmission. Due to the negative test results, we proceeded with the muscle biopsy, which showed the presence of rod-shaped nemaline bodies that are indicative of nemaline myopathy. So we really had the idea that this was the nemaline gene that we had to look at. The exome sequencing was performed at the NGS here at the SHU, which we call the next generation sequencing platform. Variants were then analyzed by our PRISME pipeline. We have identified a new homozygous mutation in intron 144 of the nebulin gene. The mutation is classified as a variant of unknown significance by the American College of Medical Genetics and Genomics, the ACMG. It was identified as non-significant or unknown significance. Mutations in the nebulin neb gene have been associated with nemaline myopathies as suspected from the patient's muscle biopsy analysis. This is what we find here. This is expressed mainly in skeletal muscle. It is closely associated with actin fine filaments, the fine filaments of actin. The C-terminal is anchored in the sarcomeric Z disc. It has the role of regulating the filament length of the actin and interaction with actin myosin, which leads to the dysfunctioning of those filaments. So that there is a dysfunctioning of muscle contraction. Nebulin is one of the largest vertebrate proteins with a molecular weight of 600 to 900 kilodalton. In humans, the neb gene is located on chromosome 2. It consists of 183 exons, of which 42 are alternatively spliced, as we called it, giving rise to multiple isoforms. Exons 63 to 66, 82 to 105, 143 to 144, and 166 to 177 are all alternatively spliced exons. Exons 143 and 144 have never been detected in the same transcript, so it's one or the other. This deep substitution located at the 118th base pair after the end of exon 144 could, according to our forecasts, be the source of a new splicing site, which would be called natural splicing. The new splicing site created by this substitution would be just as strong as the natural splicing center, and there could be competition between the two splicing sites. So both splicing sites, which we find at intron exon 144 and exon 45, might be used, and a disruption in this region could affect the level of the isoforms as well as their functions. So the sequencing of the clones allowed us to demonstrate that these additional base pairs correspond to the base pair 118 of intron 144 just before the location of the mutation. This result suggests that in this case, the new splicing site was privileged and a pseudo-exon was formed. The addition of these 118 base pairs causes a shift in the reading by changing the amino acid sequence to introduce what is called a premature stop codon. These mutated messenger RNAs are likely to become the target of degradation by what we call a nonsense-mediated decay, which is a natural degradation process of unstable RNA. Indeed, very few clones with this partial intron retention have been detected. This suggests that these isoforms are unstable. The expression of fragments 142 and 145 of nebulin in the patient is decreased by approximately 34% compared to a healthy control. So this suggests that some isoforms of nebulin are affected by the mutation in intron 144. The decrease in the expression level, but also the modification of the ratio of isoforms could disrupt the activity of nebulin. Overall, these data suggest that this new homozygous mutation in the NEB gene alters the expression levels and/or the function of nebulin in the skeletal muscle of this patient. It is therefore highly likely that this variant is responsible for this disease in this young patient. We just published this study last year, and the main author is Nathalie Laflamme, who is in charge of this project with my research nurse, Jenna. Second example, patient number 2. 5 patients coming from 3 families originally from Saguenay-Lac-Saint-Jean region. 3 of them died, one at the age of 16 months of a severe epileptic encephalopathy, another prematurely at 36 weeks of pregnancy, and another who presented a microcephaly accompanied by hypoxic ischemia. The 2 living patients had phenotypes of microcephaly, developmental delay, seizures, abnormal myelin, etc., although they had survived. Here, we identified in probins a novel homozygous pathogenic mutation in the pyridoxal phosphate binding protein, the PLBBP gene, as we call it. PLBBP deficiency is associated with early vitamin B6 dependent epilepsy. This is in perfect agreement with the observed phenotypes and clinical features that were observed in those patients. Interestingly, both living patients had a history of prenatal supplements taken by the mother to relieve nausea, and these supplements contained high doses of pyridoxine. So, according to the open reading frame prediction tool, this deletion in exon 5 of PLBBP would result in a truncated protein of 124 AAs instead of 285. So the analysis we did here is a Western blot, which is done on the 2 living patients, and this Western blot confirms the absence of the protein. Knowledge of the presence of this founder PLBBP deletion in the French Canadian population will facilitate prenatal diagnosis. In the context of a suspected PDE, treatment with pyridoxine or derivatives should be initiated, and that's what we did on those patients that survived. This is another study which we have published with our team of pediatricians and neuropediatricians with these families from Saguenay-Lac-Saint-Jean. We also published other papers on TDI in a child that demonstrated primary microcephaly. This is a case report that we published recently, and we collaborate with the team of Care for Rare to demonstrate other cases of these diseases. This is another paper which we have just published with an analysis of 2 families with a neurodegeneration mechanism. So our project will allow Quebec children to get better therapeutic treatments and will improve the clinical and psychological support brought to the affected families. PRISME will contribute not only to scientific breakthroughs about rare diseases, but also represents hopes for these Quebec families affected by a disease that affects their child. Our research team is made up of clinicians, Dr. Nicolas Christian, who is a neuropediatrician, Dr. Baibalacci, a genetician, Rachel Laframboise, also a genetician, Marc-André Dugas, who is a pediatric intensivist, and Nadi, who is our research nurse, Nathalie Laflamme, who plays a major role and who prepared this presentation. We have a whole team of informatics specialists without whom we wouldn't be able to find the variants, and our collaborators, which are the Cancer Genome Science Center, UBC, Care for Rare, and other organizations such as the CHEO Research Institute. This research project would never have been born without the foundations. As you know, rare diseases are very hard to get funded by classical organizations, as the research institutes in the health, because there's only 2 or 3 patients with those rare diseases. I convinced 3 foundations to invest in this project. There's the Marceline Jean Coutu Foundation, the Sandra and Alain Bouchard, owners of the Couche-Tard chain, and the Les Étoiles Foundation. Thank you very much. Thank you very much, Dr. Rivest, for this presentation, which was both educational and informative in terms of results. Of course, we can't present all 31 families for which you solved the cases. We would like to see other examples, including those where there's a treatment that becomes possible. And this has an important impact on patient management. By the way, for this second case with pyridoxine, you mentioned that the mother prenatally took some drugs against nausea. Can you clarify a bit more and elaborate on the impact this had? And yes, it could be such that depending on the mutation, if, for example, there is no worsening effect, well, the protein will not necessarily be truncated. But the fact that the mother during pregnancy takes a drug against nausea, for example, can foster the expression of this variant. And then that's when we can see that, well, it depends, because she had lost during her pregnancy, she had lost some children before. Is that the cause or what? We don't really know. But possibly this could be an additional risk factor so that the variant will express itself much more significantly. And then the child will be born or will not be born because the variant will be overexpressed because of that medication. So that's what can help us. It's like other children who simply, it's just the fact of taking vitamin B causes all kinds of cascading effects on the expression of the variant, and it corrects the mutations by itself. So that's why it could be very interesting. The problem we have with all that is that when we recruit families, then of course the doctors will do a series of tests, as you know, and all mother-child centers and all birthing centers. They will be referred, the child will be referred to us later. That's where we have to find quickly, can we find a variant? And is there something we can do on the expression of this variant with a drug that currently exists? The problem we have in our case is that we don't necessarily recruit people who have very severe diseases and who have died, for example. No, we recruit living patients. So we try to see with these recruits what can be done. And that's the idea behind all that. But currently, I think with the recruitment, we're currently recruiting other cohorts, and we've bought new devices to do the full genome sequencing and to see if we can't go back in time in children for which we haven't found the causes of their disease. And that's what we're doing. We're going to do this retrospective analysis. Just to be clear and not to scare pregnant women here, there's not a severe risk for all pregnant women to take drugs during their pregnancy for these diseases to happen. It's just that in the specific case of the genetic mechanism of those diseases, taking those drugs had an effect. It's just because those are very rare diseases. So we can't conclude that these drugs will cause, no. I just wanted to clarify this. You know, there are so many rare diseases that often we have one case among millions. And so we found that with this drug in particular, it could be because of that drug that the variant finally expressed itself. But it is so rare that it's one case among millions of cases, and it fell on this one. So we said to ourselves, maybe that's what made the variant be expressed abnormally, and the protein was truncated. So that's the picture. And that's why it's so difficult to come up. And the goal of our project is to create a registry, a repertory, an inventory, so that if one case happens in Japan or in China, in Europe, the person will say, oh, there's a case that occurred in Quebec City, and it comes from a Saguenay-Lac-Saint-Jean family, and it's the same case I have in my clinic. And can we find a drug for that? And apparently others apparently found something. And that's the goal of these groups about rare diseases. The goal is to create a registry so that if a person develops a neuromuscular or neurodegenerative disease, and finally all the tests we've done were negative, and there's one person who found a variant, the goal is to help this rare disease consortium. Because as I explained before, it's very difficult to get subsidies and to get grants about rare diseases because it's one case among millions. And that's why people get frustrated. That's why I started this project with foundations. So I told them our goal here in the Mother-Child Center, we have a lot of sick children. And finally, we have to try to help families, to help the people and say, listen, we really have to look into this because it's a very grave problem. And that's why doctors invest themselves very much in this project, because there's a real will to create this rare disease registry. And we're talking more and more about this worldwide, creating this registry. Yeah, exactly. Before continuing, I want to say to our audience, take advantage of this situation. We have the director of the research center of the CHU de Québec, who had just made a presentation about the latest technologies to diagnose diseases. Don't be shy to ask questions. You can write them in the Q&R, and for Anglophone people in the Q&A, if you have an English version of the Zoom, at the bottom of your screen, use this button. I don't know if you saw this recently. At the end of last week or this week in the journal Nature, there's been a very nice editorial article which calls for everyone to share, everyone around the world to share the information they have about variants. We noted that you compared with the Vancouver team, you consulted the Ottawa team, the OSHIO, this SHIO team, and there are some centralized databases, but I think the desire is that there be more sharing of data. Because as you say, these variants, these diseases are so rare that the more sharing there is with free access to all researchers, then the more cases we will be able to identify. Do you agree with that? Well, that's a bit why we had a very close collaboration with the Ottawa group, with Kim Bolcott, as you said, because the goal of that is to share information among ourselves, share patient information. And that's why we have a database, which we have in a software that we call GAC, and everything's there. The symptoms of the patient's symptoms, the variant we found, the possible treatments, proteins that are affected. You can click on this file and everything is freely available. Our goal is to create this registry and to share it worldwide. And then if we can access in return the other databases, that will help us too. That will help our physicians when they have a patient that they tried everything on, because the goal of the project is for patients that have resisted all analyses. We will do clinical genomics here. We will start this. We didn't do this in Quebec because it's very frustrating to know that we are in 2022 and not being able to do clinical genomics in our hospitals. we take the DNA and we send it to the United States. We send this to different locations who will do gene panels. We're not there now. Now we take the DNA, we send this onto the sequencer, and we do it here locally. And we're doing this now here at the show. So what we'll be able to do, you have a sick child who is very sick. We all know what he or she has. We do a blood sample immediately. We take the DNA, we send it to sequencing directly here in the room besides here. 2-3 days later, we come with a data pipeline. We can see what are the variants. And that's where we're at in 2022. That's what we've been waiting for a number of years, and that should start this year. It's the Quebec Molecular Diagnosis Network. Is that it? Yes, you have a role to play in this molecular diagnosis network. And I would like to say to our people, to the people who are hearing us, that what you're doing right now is essentially research. You with your team, with a research team in Ottawa and Vancouver, that's for research. So families have to be recruited for research. It's not a test that is common in the clinics yet. No, it is not. You're right. Do you have any hope? No, panels were already a progress, right, to identify undiagnosed people. But do you hope that soon in 2022, maybe in Quebec, we will accept exomic sequencing, at least exomic sequencing, for clinical cases, for any person that doesn't have a diagnosis? Can we hope for that? Can you hope for that? Currently, in 3 weeks from now, we will start clinical diagnosis here at the show. So we've totally ready. We've developed gene panels, which we're now doing ourselves. The problem we had with that is to convince the government that we were able to do it. And it was very hard to convince the government that we could do it. But now they got it. They understood. We spent millions and millions in tests outside of the province, while we have the expertise to do those tests here. What frustrated me in the project personally is that I couldn't inform the families because it's a research protocol. And I couldn't say this to the families because I had sequenced this on my sequencers in my lab. And I was so frustrated. I went to see the doctor. I said in hiding, look, the variant is this, find this clinically and inform the family. It's not logic that we had to do this and hide and tell them that's the variant, prove it clinically, then inform us. We should not be there in 2022. Secrets like that. We should sequence it all ourselves, and immediately when we find something, we inform the families, we inform the treating physician if there is something that exists to cure the child or care for the child. We have a person who is writing to us, who we know at the RQMO, who is recruited in your research project, and nothing has been found so far. As we saw, you recruited 135 families and you solved 31 cases. We know that you can't solve 100% of the cases. So that person is asking, in future, will there be something else, that some other cases solved? I think we're there. You did the exomics, there's the genomics, and there's the RNA sequencing. There's other genetic tests. Will you go to those new technologies in families for which we haven't found anything? We get back in time because we have the cells. We still have the cells. We have the DNA. We have everything. And now there are new technology. We just bought a new device, which is extremely powerful. It's a new device that allows us to get back in time and do the full genome sequencing, which we didn't do before. It's a lot more complicated doing the variant analysis with the full genome, but we're there. So we want to go back in families where we haven't found anything to see if we can't develop a new technology to get to find variants that we missed with some new technologies and the new generation sequencing with what we call the fruitinome segments. We didn't do before. We didn't have the machine. Now we're all equipped in the genomics center to do that. And we're currently developing what we call the data pipeline for variant analysis with these new technologies. Well, that's encouraging. It's encouraging to see that there are breakthroughs or progress made. Just one last question. There's not a lot of time for questions, but there's a question we get very often, and it's sometimes difficult to understand by the families. You spoke about the VUS, which are variants with uncertain signification, uncertain meaning. Why do we say that these are uncertain? So we can't say that this is the cause of their disease. Could you just quickly explain this so that our families understand better? The VUS, actually, there's so much instability that it doesn't allow us to predict 100% whether or not this variant will cause a mutation in an amino acid. And that's the problem we have with those, because it could be a point mutation. But we absolutely need the variant to be associated with a protein dysfunction. Otherwise, it's useless. And this variant has to cause a protein dysfunction. Otherwise, there's not going to be any change. Everybody has variants, everybody has mutations and so on, but it doesn't mean that it will lead to a truncated protein, a mutated protein, or a dysfunctional protein. And that's where the depth of analysis and genetics comes into play. That's why we take the DNA and we take the proteins and we do the whole range of biochemical analyses to see what's happening. That's what we did with Nebulin to demonstrate that this is a real effect on proteins. It was extremely narrow on exon 144. That's why when we published, the arbitrators in the review panels asked us evidence that things were modified. We proceeded with muscle biopsies to show all the analysis that was insufficient at that time with this small and non-functional amino acid. Well, it's a good thing you explain things very well, because the thing is that when families receive genetic test reports and they see the VUS, they do all kinds of research on those genes. It's okay, it's good that they do research, but they have to be told clearly that you're doing all the work to prove whether or not these genes are, maybe one day you'll prove that one VUS is the variant in question. We have to stop there, unfortunately. I just want to say that our month of February is continuing. Actually, the next Zebinaire next week will be with your colleagues at Care for Rare, Dr. Rivest. A researcher will come to us in the Care for Rare group in Ottawa. She will be making a presentation on this pan-Canadian project that you're part of. And once again, other gene discoveries were made in Canadian families. And then we will have two more Zebinaires, one by the president and founder of Genetic Alliance in the United States, and one last from Montreal UPNIVO about our project in collaboration with the RQMO and a whole lot of other people in life sciences to devise a strategic plan for research on rare diseases. And you will see that your questions and comments about the funding of research on rare diseases is shared. Registries, everybody speaks about that. We are proposing a lot of things to our government to solve this issue of registries. And of course, on Saturday, we have our Génie en Rare, our contest, where students of medicine will take part in a contest to answer a number of questions about rare diseases, not just specific diseases, but the rare disease approach and what patients experience. That will be all day Saturday, always virtually, and on February 26th, our day of education about diagnostic wandering. How can we better support people? We'll have testimonials and how to bring the physicians to think rare. Thank you very much. And I thank once again Mr. Rivest for taking this time to explain to us this nice project, and our sponsors who make possible all those activities during this month of February. Thank you very much. So long. Have a great day.